cycle is a series of events that cells go through as they grow and divide. Stages of cell cycle are interphase. There's 3 stages. We'll go to that later. And then also mitosis and cytokinesis. Mitosis also has 4 different stages. We'll go through that later. Gotcha.
Here we have a chromosome. Starting in interphase in G1, the chromosomes are a single chromatid, as shown. During S phase, the chromosomes replicate and form 2 sister chromatids, also as shown. And at the end of interphase during G2, the chromosomes prepare for M phase, or mitosis and cytokinesis, which is the completion of the cell cycle.
As you can see, we have a cell, and this is the beginning of mitosis in prophase. In prophase, you can see the chromosomes which are visible, because all the DNA condenses and makes chromosomes. And the spindle fibers, which are the things on the outside, start to form. And that leads us straight into metaphase, which the chromosomes line up in the middle, and these connect to the center of the chromosome.
So that is the end of metaphase, and the next phase in the cell cycle is anaphase, which is where the chromosomes separate and move along these spindle fibers that they are currently attached to the opposite sides of the cell. And that is the end of anaphase, and now we're moving on to the final phase of the cell cycle, which is telophase. And so in telophase, the nuclear envelope, which I'm drawing currently, forms, and that is telophase, and so now we are ready to move right along to cytokinesis.
This stage is the final stage of mitosis and interphase. It's called cytokinesis. In this stage, the cytoplasm pinches and comes in half. We'll demonstrate this but this only happens in animal cells. In plant cells, something different happens. As the cytoplasm splits, they go their own ways. But for plant cells, there's something called a cell plate that comes in the middle because they have cell walls. And then it comes out and then splits it apart, just like that, instead of pinching the cytoplasm.
The next topic is meiosis. Meiosis is the process of making sex cells, which involves 2 nuclear divisions. Meiosis I and meiosis II. This occurs in the gonads, or the testes or ovaries. Sex cells divide to produce gametes, sperm or egg, and the gametes contain half of the number of chromosomes or body cells, which is the number of 23 instead of 46. And 4 daughter cells are created in mitosis instead of just 2. In meiosis, there are 2 divisions of the cell. In the first division, it begins with prophase, which the homologous chromosomes in the nucleus begin to pair up with one another, and after that, they split back into chromatids, or just 1/2 of a chromosome, where crossing over can occur, which could increase the genetic variation of each chromosome. The second step of the first division is metaphase, which is where the chromosomes all line up at the equator, or the center of the cell. And then the sequence of the chromosomes is lined up at random, which is through chance, so that also increases the genetic variation. And then anaphase occurs, which is where the homologous chromosomes move to the opposite poles from the equator, just the opposite sides of the cell. And then lastly is telophase, which is where new nuclei forms near each pole alongside its new chromosome complement. So there would just be a split in here, creating 2 new cells. And then, during the second division, the nuclear prophase 2 is how it starts, and the nuclear membrane disappears, and the second meiosis division is initiated. Then, during metaphase 2, the pair of chromatids line up at the equator of each of their cells. So you can just imagine there are two cells right here, and they're lined up at the center. Then during anaphase 2, each of the chromatids pairs move away from the equator of the poles using the spindle fibers just in mitosis, just like that. And so now they're on opposite sides of the cells that they're in, and there are two cells. And then during telophase 2, 4 new haploid gametes are created that will fuse with the gametes of the opposite sex to create a zygote. So after that, each of these cells split, creating 4 different cells, and all with different genetic material.
As a final result of mitosis, 2 cells are formed. These 2 cells are genetically the same and are reproduced by asexual reproduction. The type of cells this process occurs in is somatic body cells, and the number of times these cells divide is 1.
So, as you can see here, we are at the end of meiosis. 4 cells are produced at the end of meiosis, and they are all genetically different. And the reproduction of the cells are sexual, given that the cell occurs in the reproductive system.
Along with these 2 types of cell division, they both have many similar qualities and different qualities. Similar qualities are they both happen in eukaryotic cells, and also they're both a type of cell division. Also, 2 differences are meiosis creates 4 reproductive sex cells, while on the other side mitosis creates 2 somatic body cells. Another difference is mitosis is asexual reproduction, while meiosis is sexual reproduction.
The purpose of mitosis is so that, 1, we can continue to grow, because without mitosis our cells would not keep forming, and then we would have the same amount of cells that we would when we were a newborn, which would mean that we would all be the same size, so we wouldn't be any bigger. And there are also, because of mitosis, we're able to heal all of our wounds, because if we got a huge cut on your arm, like Mr. Books has said, there would be no way to fill in the gap with all just no cells. I don't even know what would happen without mitosis. We would be lost without mitosis.
The purpose of meiosis is so we have an abundance of sex cells so we can reproduce, and also so we can all look different and have genetic variation. Go. It has been a pleasure for you to join us in the learning of mitosis and meiosis. Thank you.